And we are live here at the ROM on our, I think it's our 4th Google chat here at the ROM. We're here today with Dr. Claire Healy and Maureen Zabowski from the Invertebrate Zoology Department. Before we get into it about octopus, snails, mollusks, and parasites, I want to give you a bit of a heads up on what this is all about. So again, if you're on Twitter, follow along on ROM Hang, hashtag ROM Hang. And what we're trying to do here is create a conversation with our curatorial team, our back-of-house team, for you. So we have programs here at the museum that expose our curators in some ways. Curators Corner happens last Saturday of every month. But really, we have more demand than we do have programming time. So Google Chats is a new platform we're trying, where we bring out some of the folks from behind the scenes. Just a half an hour. We'll be here for a half an hour. And we ask them some questions about their research, about their collections, about their personal lives, about their jobs, and then what's on top of mind, and ask you to join in and follow along. So that's kind of the plan today. I'm just going to, this is typical of it, but Ryan, there's someone at the door. Maybe you could go have a look. Hi, Maureen.
Hi.
Hi, Claire.
Hi.
How's your day going today?
It's great. Lots of wintry weather. Mixing in with our, April showers.
It's crazy. It was hail this morning.
Yeah.
Welcome to Toronto. We did have someone tuning in from Georgia last time. So for those in the international audience, there was hail in Toronto this morning, quite substantial. Maureen, I'm curious. You're a technician here at the ROM. Can you describe to us what a technician in the Invertebrate Zoology Department at the ROM does?
A technician does a lot of work with collections maintenance, a lot of sorting, identification, databasing, labeling. And one of the collections that I just recently finished was soft coral collection that was donated to us, and it's a very diverse collection. The soft corals are very good part of the coral reef ecosystem. They provide habitat for other invertebrates and many different types of fish.
So quick question for everybody out there. First of all, just speak up a little bit more and tell us what a soft coral is. I mean, the general folks out there think of a coral as a tree-like looking thing that sits on the ocean floor. Is that what a soft coral is?
It can be. There's different types. There's the stony corals, which are the heavy calcium carbonate base to them, and then there's the soft corals, which are more flexible and composed of a different structure. And they tend to be the fan corals.
Okay.
If you know the purple fan corals, those are a very common one.
And aren't some of these soft corals found at like extreme depths as well?
Various depths.
Yeah.
Yeah, it can be.
So how many corals from this collection that you just went through, how many of them would be extirpated or extinct or no longer on the planet? Any of them, or are they mostly?
No, they're still all, as far as I know, extinct. So
Maybe we can come back to that one, because I know there is a global issue with some of our coral reefs. But before we get to that, Claire, Associate Curator of Invertebrate Zoology here, and we're going to play a quiz show with you later on something that has to do with your research. I'm really looking forward to that. But tell us what an Associate Curator is, what you do on a day-to-day basis here at the ROM.
Well, I'm a researcher. So in addition to overseeing the activities within the collection and maintaining the running of the collection, I run a research program, and my particular expertise is in parasites that live in the intestines of sharks and rays, at least for part of their life cycle, for the adult portion of their life cycle. And so I do a lot of taxonomy, which is describing and classifying species and other higher level groups, as well as systematics research, so expanding our understanding of the evolutionary history of those animals.
And just like our terrestrial ecologists who study polar bears and big cats and elephants, the connections in life at that level that you're talking about, the parasites of sharks and rays, these are small organisms. They're just as connected to the tree of life as a bear is, right?
Absolutely. So if you look at the tree of life of those worms, they actually evolved, at some levels, they're related to free-living worms. So yes, everything is related evolutionarily, but also just ecologically. We're all connected in these complex networks we call food webs.
Yeah.
And so they're definitely an integral and very underappreciated component of food webs. So they're really integral in a lot of different ways, and often overlooked because they're in such an unusual and out of the common sight of people. So they're outside of our usual thoughts about the ecosystems that we encounter, that we rely on for our sustenance and our health and the world's health, etc. So.
And it's a really important point because people, when they consider the word biodiversity, we've got a banner out front this month. We've got a huge web presence, but we still find through some of my colleagues in the education sector that the term biodiversity is poorly understood.
It's still jargon. Yeah, it's definitely still jargon in some cases to many people. And I hope it will get beyond that point, because I think if we just use the term diversity, that has such broad connotations in our English language, right? So, and I think with greater understanding of the importance of biological diversity, that people will sort of slowly allow biodiversity to trickle down and become a sort of normal, expected, non-jargon component of language, so that people recognize sort of the stature of the importance of that concept to all of us. And I left out a couple things that I do as a curator, and one is teach at the University of Toronto, where I'm cross-appointed in the Department of Ecology and Evolutionary Biology. So it's nice being able to have that relationship with students and academics. And also the Shad Gallery of Biodiversity and other galleries and exhibitions within the museum, I'm invited to and help in many cases produce those, if my expertise will lend itself to the project.
And I want to get into a Curator's Corner we did last year. Quickly, Maureen, the Curator's Corner we did last year, were you involved in that?
I was involved in one, cephalopods.
Yes, with the game where we had the predator-prey.
No, that was...
Okay, well, I'll talk about that in a second. But the segue quickly is, how many individual species are in our Invertebrate Zoology collection here at the ROM?
I honestly couldn't tell you.
Take a guess.
Too much.
Species?
We talking 10,000? We talking a million? We talking...
Oh gosh. Okay, so the invertebrates are in a museum. They're all the animals. Well, just philosophically, invertebrates are all of the animals that lack a backbone. So incredibly diverse. But because of the enormous diversity of insects within a museum, just practically speaking, we have experts only on insects. So the collection that we take care of excludes the insects, and it's everything else.
That's where I was going.
All the worms, all the sponges, mollusks, cnidarians, so jellies, cephalopods, etc., all the way up to echinoderms, etc. Starfish, sea cucumbers, all of those wonderful animals. So we know that our collection includes about 1.2 million specimens, which is an enormous amount of specimen diversity. We know that virtually all of the roughly 33 major lineages of animal life, invertebrate animal life, so phylum level diversity, virtually all of those are represented in the collection. And we have specimens from all continents and all oceans on the planet. So it's an incredibly diverse collection. But in terms of numbers of species, many, many thousands.
Many, many thousands.
I don't think we can, a lot of the, many specimens, entire collections will come to us not identified beyond the level of family, right? Or there'll be environmental collections where you'll go dredge a chunk of sediment, say from the deep sea or from rivers in Ontario, and you'll deposit it in toto, right? So just whole for people eventually to get to and work on as a historical reference of what was there at a particular time.
Are we going to get access to Jim Cameron's dredging of the Mariana Trench?
I don't know.
Well, we could pull some strings
maybe
for you.
Yeah.
So Maureen, you're one of the best behind-the-scenes tour guides I've ever seen at the ROM. You're a pro at it. And that's why we're taking EVC behind the scenes with you again. I mean, it's great. But you have some favorites, right? You go behind the scenes. You know what your audience wants to see. And it's not necessarily a bunch of little things. So what are your 2 or 3, and you've brought a few things here for us today. What are your favorites to show guests?
I find the mollusks have become my favorite group to look at and study. I spend a great deal of time organizing and identifying and putting them away, making sure that everything that comes in contact with them is acid-free. As you know, anything acidic will erode the shell. So,
hey, I bet you 9 out of 10 people didn't know that.
So all the cabinets, the containers, the batting, everything has to be acid-free down to the paper. So I had to do all that, and in doing that, I probably saw every single specimen in that collection. So
1.3 million.
Of just the dry mollusks.
Okay, okay.
Actually, I think we have about 10,000 dry mollusks, or more, because they're not all in the database. But after I did that, I gained an appreciation for their color, beauty, and diversity. So
can we show some people?
Yeah, sure.
You have to look at the camera here just to really see what they can see.
So I brought, this is called a golden cowrie.
Ryan, show that off for us. Be very careful.
Very beautiful. And it's quite harmless. It's a grazer, so it feeds on algae. So there's nothing.
It's nice to turn it around.
Oh yeah,
The animal.
The animal will.
There.
Where's the animal,
It's not
Maureen?
there now,
Okay.
but it produced the shell. So that's what's amazing about these animals. They produce such
They're artists.
beautiful shells, yeah. And this one, well, this is a very intricate design. That's a cone shell. All cone shells are capable of producing poison, and some are more deadly than others. And this one looks pretty harmless, but this is the most deadly cone shell of all.
Whoa, cool. We've got something cool here, Ryan. The deadliest cone shell. What's the proper...
What happens if I touch it? Am I going to be okay?
The animal's gone. It's from the animal.
Hi, guys.
So there's a proboscis, and it feeds on fish. So, and it feeds at night when the fish are sort of resting and not aware of what's going on. And it will shoot this harpoon hollow tooth into the fish with the venom, and that immediately paralyzes the fish. And then it engulfs the fish with its proboscis and digests it, brings it in, and then when it's done, it spits out all the bones.
Wow, that is incredible. What kind of depths is this thing living at?
It's not that deep. That's why, and a lot of people look at it and go, that's beautiful. They'll pick it up and they'll get the venom.
And what's the reaction to a person from a bite or from a sting?
Bad. There have been 30 deaths from this particular species.
What a great story. Okay, it's quiz time here on the ROM's game show, ROM Hangout. Dr. Claire Healy. Before we get to the game show, Claire and I and Maureen and a ton of other people here at the museum did something last year where we tried to frame Claire's research. And again, this is my perception, my understanding of her research. She studies the parasites of sharks and rays in marine environments. So these are tapeworms and other very small invertebrates that live inside a host, a shark or a ray, and need that host for their entirety of their life cycle. Is that about right?
Yes.
And the diversity and the evolutionary biology of those critters. So kind of an esoteric bit of research. And so what did we do last year, Claire, in April of 2012 to help promote that research and promote Canada's oceans and oceans environments in general? Maybe you could tell us about that project and then we'll do the game show.
So I presume you're talking about Canada's Oceans in View.
Yes.
Okay, so we produced a lovely exhibition that covered the diversity and sort of the importance of the oceans around Canada to the people of Canada and to people in general, of course, and all oceans to all people is what follows, I hope, upon all of that. But what we did is, my small aspect within that exhibition was just to highlight the research that I do relating to sharks and the shark diversity and elasmobranch, or shark and ray diversity around Canada. Canada has 3 oceans around its coast, an enormous coastline, and we have a lot of uses for the organisms that live in the ocean. And so people hopefully came away from that exhibition, which highlighted each of the 3 oceans, many of the organisms, and their interactions within those ecosystems, and then how people relied on all of those. But one of the neat things that we produced, I think, from that exhibition was these very complex summaries of food webs in each of those oceans. And one of them was where we started focusing on the top predator being an apex predator shark, so a very large shark. And we worked down with resources from the primary literature all the way through all the other kinds of tertiary literatures that we could get our hands on to really fill out in some detail the complex food webs. And one of the things I used those renderings of those food webs for later is I sort of marked on them casually all the places on those food webs where parasites occur, right?
Oh.
Multiple kinds of parasites on each of those levels of the food web, just to show people who visit my lab and who want to know more about research relating to parasites. How do they fit in? Because when we do think of food webs, we really do think of human mouth. Where does that lead? It usually leads, in the ocean, a fish, okay, and that fish eats a copepod, and that copepod might need some algae. But in other words, in the common person's, in every person's mind, it's very simple. One linear line, right? When you fill it out for people, what actually is the range of the diets of all those organisms and how are they interrelated? Even then, when you're sort of comprehending at that level, you're not thinking of the parasites of all those organisms, are you? And yet they're at every level. They have an incredible impact at each of those levels, and it's much more interesting. It's a much more accurate picture if you do think of it in that way.
Accuracy is the big one, the takeaway from that, I think. So during that project, and following that project, the program here we run at the ROM called Environmental Visual Communication, which is a program where we try and bridge the arts and science and build a really good storyteller out of a graduate student who has a little bit of a sense of environment and a little bit of sense of videography. And so what they produced for Claire are some cards. These cards, and I'm just going to get up, you'll hear me, but you won't see me. These cards have lovely pictures of...
Turn it up. Okay.
Yep, I got that one.
A common mistake.
So this is a very blown up macro shot of an animal, a parasite of a shark or a ray that Claire has identified out in the field. And I know the answer on the back of this card, what species it is. And I've got about, 6 of them or something. I'm going to show these to Claire, and we're going to give her 10 seconds to give us the name of the animal and what shark or ray it came from. We'll see how she does. You ready? Claire, are you ready?
I'm ready. Okay, so the one you just saw is Rhinobothrium macrocanthophallus.
What the?
And yes, and it's from the giant freshwater whip ray from Borneo, though. So, and that's Hymentura polylepis. I describe this as a new species, so I'm intimately, aware and knowledgeable of this one. This one's too easy.
Okay. Next one.
But it's a very nice thing.
This is my favorite. I've put it into a PowerPoint presentation that I delivered at a keynote yesterday. And Claire is going to see it again for 10 seconds.
Okay, this is, I'm sorry there are no common names for these. It deserves a common name.
It does!
I think. But it's hard enough coming up with a new name. So I actually named this as a new species as well. This is Platybothrium tantulum. And I named it tantulum because it is so small. That's what that means. And I love it too. It's actually an interesting story. This one is from hammerhead sharks.
Hammerhead sharks?
It's from, Spirna zygena and Spirna lewini, which I prefer to call luini. And so, unlike many of the tapeworms in the various orders that use sharks and rays as their hosts, so many of them are host specific. This guy, though, or gal, because they're hermaphrodites, this species actually uses 2 hosts as its host. It can be found in either of those two species of hammerheads. I discovered it off the coast of the United States, the eastern coast of the United States, in the Atlantic Ocean, in those hosts. And what's interesting then is, so I based my understanding of that host diversity, that host distribution, just on the morphology of the worm. I thought they looked the same, and I described it as a new species, etc. Later, a colleague of mine actually collected molecular data that then supported my claim that this was one species found in two hosts. So that was a very nice sort of example of where sometimes, when you're lucky, perhaps, or just when things work out right, you actually get later independent verification of one of your findings, because every species description is a hypothesis, right? Anyone else can go out and look at the organisms, their behaviors, every other type of information you can collect about them, including their DNA, and
challenge you.
See if that, well, yeah, see how that evidence lines up with the previous hypothesis. And so that has a little story.
All right, I got to find a trickier one. She's too good here. Ooh, yes. I think we have a stumper, folks. Check that out. That is incredible. Claire, tell us a little bit about the photography on how this was created before you ID this one. How did we get these incredible photos?
So scanning electron microscopy has gotten a lot easier because we have very sophisticated microscopes now that do their work in a very quick way, and because over many years of experimentation, biologists in various labs have worked out optimal ways of fixing or killing the animals so that they can be preserved over very long periods of time in museums and other collections and still be useful for techniques like this. Scanning electron microscopy allows us to look at the fine structures on the surfaces of these worms. This is just like the anterior end of one particular tapeworm. But if you zoom in on any region of that worm, they're actually covered in minute surface structures that are unique to tapeworms. And so we use scanning electron microscopy really routinely. And
the species and the host?
This is Crassoseptum pietrofaciae, actually. Some colleagues of mine and I, Florian Reda, and a student of his, and myself, described this as a new genus, actually. Yeah, so, and it's found in Mobula coolii, this fantastic ray that we were able to examine off the coast of Borneo.
Cool.
And so it's an entirely new genus. So far, it's the only species known in that genus. And not enough work has been done, actually, in Mobula and all of its devil ray or bat ray relatives as far as tapeworm diversity. Some work has been done, but not very much. So I encourage everyone who's interested in examining tapeworms to go and look at Mobula and its relatives for tapeworms.
Maureen, how do you get involved in the collections and the storage of these incredibly small organisms?
Yeah, it's
challenging?
It's very different. Yeah. First, we have a whole different way of storing and databasing. So,
and what is that?
Well, you've got your tissues from the worms themselves. You've got tissues from the host. So you've got.
Are they on slides? Are they in drawers?
The tissue is stored in 95% and then stored in ultra-cold freezers.
So little jars of alcohol.
Yes.
Okay.
Either usually the whole worm. Tissue.
Okay, guys, we have 5 minutes left. Again, we're here at Google Hangouts at the ROM, a new program we're doing to try and expose our curatorial team, our back-of-house team, to the public in a new, free way. We've got a neat question coming in from Twitter here, which we're really encouraging. Stacy. Hi, Stacy. Stacy Kerr.
Oh!
She's watching. Hi. We know Stacy. She's a former student of ours and a former student of the EDC program. And Stacy writes, hi, Claire. Are there species of sharks or rays that have more invert parasites than others? Does habitat or depth matter?
There are definitely species of rays and sharks that have lots more diversity of parasites, particularly tapeworms, than others. The most species of tapeworms I've ever found in a host was in Tinea limma, this beautiful blue-spotted stingray. And I think it's 25 species. Actually, this 25 species of tapeworms or so, and it was the same for the giant freshwater whipray that we examined from Borneo. This animal and others of its species hosts, the host, the ray that this worm comes from, hosts, in addition to this worm, about 20 other species of tapeworms. So that's an incredible diversity of tapeworms in one animal.
Do they compete amongst each other for the same resource? They must have niches that they occupy then, or is it complicated?
It's complicated, but people actually are addressing that issue. We basically don't know enough to say a lot about those sorts of questions because we're still really doing a lot of discovery. But we can definitely say that a lot of deep sea sharks and rays are parasitized by fewer tapeworms. There are also particular lineages of sharks, for example, that have, to our knowledge so far, very few. They're just depauperate in terms of their tapeworm parasites.
Cool. Interesting little comment from Christy Van Hoven from Twitter. I've got to get to one other thing, so this will be the last question from Twitter. But Christy says, diets of parasites, sweet. So what are the parasites? I think she's asking, what do they eat?
So if we're talking about tapeworms, it's really they live in the gut of their vertebrate host, and they absorb through their tegument surfaces very small molecules.
Just like us then. And
no,
wait.
We engulf our food and swallow it.
But don't I have
complex
tapeworms?
digestive system? They have no digestive system whatever. So they're absorbing very small molecules, and that's how they derive their nutrients. So they're stealing those nutrients from their host digestive tract. Now, if you're talking about all sorts of parasites, of course, yes. We've got parasites of the external parts of our body, the internal parts of our body, just like every other vertebrate animal. And depending on the kind of animal you're talking about, parasite animal, they're going to have a various, huge diversity of feeding strategies. Everything from leeches and other blood-sucking animals, to animals that live within our blood cells, like malaria, etc. So, in other words, as diverse as you can think of parasite, they're going to have a diverse array of feeding strategies.
Cool. I want to wind this down, guys. We have a minute. We could have talked for another half an hour, I think. But I definitely wanted to finish on a note around oceans. So Claire and I have been emailing back and forth over the last couple days around a new book that's coming out called Jellyfish. What's it called?
Stung.
The role of jellyfish in the environment. Quickly prefacing it, maybe Ryan, you can show this. This is a curriculum-linked, Ontario curriculum-linked guide to students in grades 4, 5, 6, 7, and grades 10, 11, and 12 that the ROM built out for the province of Ontario. It shows all kinds of ways that the student in Ontario, and believe it or not, Ontario has an ocean, how they can take the curriculum standards that they're given as teachers and include ocean-themed subject matter into the learning process. One very strong theme throughout this curriculum package, which is available on our website at ROM Toronto, is the concept of jellyfish taking over the world. What's that all about? Quickly, Claire.
Jellies are incredibly adept at taking over where a gap has appeared in a food web. So if you have a diversity of animals, a diversity of food web niches in a community, and suddenly some of those populations are becoming very weak, very small, and the former, job of those organisms in that ecosystem is being left empty. Jellies are incredibly adept at taking over those niches and taking up that space and just feeding and feeding and feeding. They're amazing predators.
So
I don't think of them as predators.
So I'm going to pick away at one point you made. Why are some of those niches in our oceans being left empty?
Because of the incredible overharvesting that a lot of, the pressure of the human population and it's putting on all of our food resources, but definitely including the ocean's resources.
Yep.
Of course, there are also other issues with pollution and nutrient loading in those ocean ecosystems that lends itself to this kind of ocean bloom of jellies and other stinging animals. But
interesting. So we're trying to figure out if we can get the author of Stung into the ROM maybe sometime this year to talk about that book. But if we're unable, again, the book is called Stung. The author is
Lisa Ann Gershwin.
Lisa Ann Gershwin. And again, our curriculum guide for understanding our oceans and how it works with our curriculum here in Ontario is available from the ROM website. Maureen, Claire, thank you so much for hanging out today.
Our pleasure.
Awesome. Bye.